Dear students, in the last class we discussed the double helical structure of DNA molecule. Today I would like to discuss packaging of DNA helix. So we know that the DNA is a long polymer of deoxyribonucleotides. The DNA is very lengthy. How this lengthy DNA can be present within the small cell? That's the thing that we are going to discuss in this video. So the cell is very small. How this lengthy DNA can be present within the cell? Definitely the DNA should be packed. If the DNA cannot be packed, what happens? The DNA cannot present within the cell. The cell cannot hold the DNA, so that the DNA should be packed. So what is the reason of the packaging of DNA helix? If the DNA cannot be packed, that cannot be present within the cell, or the cell or the nucleus present in the cell cannot hold the DNA, because the DNA is very lengthy. So the DNA can be folded in a proper manner and it should be present in the nucleus. How it happens, that we are going to discuss. So the distance between the two base pairs. So we know that the double helical structure of DNA will be like this. So here the bases will be there, A, T, G, C, like that. A binds with T, A, C. So the distance between the two successive base pairs is 0.34 nanometers or 0.34 into 10 power minus 9 meters per base pairs. So this is the distance between the two successive bases. We know that how many base pairs are present in the human diploid cell. The number of bases present in the human diploid cell is 6.6 into 10 power 9 base pairs. So the length of the DNA can be calculated from this. So the length of the DNA of human that is diploid is 6.6 into 10 power 9 base pairs into 0.34 into 10 power minus 9 meters per base pairs. So if you calculate, it comes around 2.2 meters approximately. So how many meters? 2.2 meters. So what is the size of the nucleus? 6 micrometers. So 6 micrometers is the nucleus. The size of the nucleus is very small. What about the length of the DNA? Very long. That is 2.2 meters. This is micrometers. DNA is present within the nucleus. The size of the nucleus is nucleus size 6 micrometers. How this lengthy DNA can be packed within the nucleus? So the lengthy DNA should be folded many times, many times in a proper manner. Then only the folded DNA can be present within the nucleus, or the nucleus can hold the folded DNA. Otherwise, the DNA cannot. If the DNA cannot be folded, what happens? The nucleus cannot hold the DNA molecule. So the lengthy DNA should be packed. If the DNA cannot be packed, the nucleus cannot hold the DNA. So that the DNA must and should be packed. Now packaging of DNA in prokaryotes. So you know that the prokaryotes usually consist of the nucleus which is not surrounded by the nuclear membrane. Truly speaking, we should not call nucleus, it is a nucleoid. The nucleus is not surrounded by nuclear membrane in case of prokaryotes. Though the nuclear membrane is absent in prokaryotes, the DNA cannot be scattered in the cytoplasm or cell. The DNA should be confined to one particular place. How is it possible? For example, this is the nucleus. The nucleus is not surrounded by nuclear membrane, no nuclear membrane. Here what happens? This is the cell. Here the DNA should be present here and there, and it is not scattered throughout the cell. The DNA is confined to one particular place. How is it happens? So the DNA should be packed in the prokaryotes also. How the DNA can be packed? You know that the DNA is negatively charged. DNA is negatively charged. So which imparts negative charge to the DNA? Usually, you know that the DNA is made up of long polymer of deoxyribonucleotides. The nucleotide is mainly composed of three components we already discussed: sugar, nitrogen base and phosphate. Yes or no? So phosphate is present in the DNA. Due to the presence of phosphate, that phosphate can impart negative charge to the DNA molecule. So why the DNA is negatively charged? Due to the presence of phosphate. So DNA is negatively charged. So the negatively charged DNA can bind to the some of the positively charged proteins. Positively charged proteins. So here some of the positive proteins can bind to the negatively charged DNA. What is the type of the protein here? Histone-like proteins. They are not histone proteins. Very, very important. In prokaryotes, histone proteins are absent. But histone-like proteins, normally called HU proteins. So these HU proteins are positively charged. So the positively charged HU proteins can bind to the negatively charged DNA. So that what happens? The DNA can be confined to one particular place in case of prokaryotes. Now DNA package in eukaryotes. Now coming to DNA package in eukaryotes. Like there are the prokaryotes, in eukaryotes also the DNA should be packed within the cell. So in case of eukaryotes, what happens? The nucleus usually consists of nuclear membrane. So the DNA should be present within the nucleus. Here also the negatively charged DNA can wrap with the positively charged protein, so that the DNA can be packed, or DNA should be confined to one particular place. For example, this is the cell and this is the nucleus. The nucleus consists of nuclear membrane. So here what happens? The DNA should be present here and there. How the DNA should be confined to the nucleus? The positively charged proteins present in the eukaryotes called histone proteins. The special type of proteins present in the eukaryotes called histone proteins. Histone proteins are present only in eukaryotes, absent in prokaryotes. Histone proteins can bind to the negatively charged DNA, so the DNA should be packed within the nucleus. So histone proteins. Now we will discuss a little bit about histone proteins. Histone proteins are positively charged proteins. And they are basic proteins. Histone proteins are positively charged proteins and basic proteins. Okay. Now, why they are called positively charged proteins? So, or why they are called basic proteins? They are basic amino acids. You know that protein is made up of amino acids. Amino acids are the building blocks of proteins. So histone proteins means that definitely they should contain amino acids. So there are two special amino acids present in the histone proteins. They are lysine and arginine. There are mainly two types of the amino acids present in the histone proteins. They are lysine and arginine. Both are basic proteins, basic amino acids. So that's why due to the presence of lysine amino acid and arginine amino acid present in the histone proteins, these proteins are called basic proteins. Now coming to the positively charged positively charged proteins. Why the histone proteins are called positively charged proteins? Because of the presence of two amino acids, same lysine and arginine. They impart positive charge to the histone proteins. How? This lysine amino acid and the arginine amino acid usually contains the side chains. You know that amino acid structure, amino acid consists of side chains. Due to the presence of those side chains, normally that side chains contains the positive charge, which imparts positive charge to the histone proteins. That's why histone proteins are positively charged proteins and they are basic proteins because of the presence of basic amino acids, lysine and arginine. Now, you know that the DNA is negatively charged. Why? Due to the presence of the phosphate group, the DNA is negatively charged. The negatively charged DNA can bind to the positively charged histone proteins and forms a structure called nucleosome. So, negatively charged DNA binds to positively charged proteins, charged histone proteins. Normally histone octamer. 8 histone proteins forms the histone octamer. That we'll discuss in detail later. Histone octamer and forms a structure called nucleosome. So what is a nucleosome? The negatively charged DNA binds to the positively charged histone proteins or histone octamer and forms a structure called nucleosome. How the nucleosome looks like? So we'll depict the picture of nucleosome. Now coming to the structure of the nucleosome. How the nucleosome looks like? Usually the nucleosome looks like this. This is the histone proteins. The H1 protein. This is H1 protein. This is nucleosome. Structure. So the nucleosome is nothing but the negatively charged DNA can wrap the positively charged proteins and forms a structure called nucleosome. So this black color structure represents the proteins, histone proteins, and this blue color represents the DNA molecule. And this is the H1 protein. So normally there are 5 different types of histone proteins are there. 5 types of histone proteins are present in the nucleosome. So what are those 5 different types of histone proteins? H1 protein, H2A, H2B, H3, H4. So total how many proteins? 5 proteins. So what are the 5 proteins which are involved in the histone octamer? H1, H2A, H2B, H3 and H4 proteins. Among these, H1 protein usually links the 2 nucleosomes. This is one nucleosome. Another nucleosome can be linked. 2 nucleosomes can be linked with the help of H1 protein. Histone octamer. So, in the structure of the nucleosome, normally 8 histone proteins are present. That is, for each 2 copies of H2A, H2B, H3, H4 can combine together to form the histone octamer. That we will see now. So, what are those histone proteins? This is H1 protein. Now H2A, 2 molecules. H2B, H3, H4. Total how many? 8. So 2 molecules of or 2 copies of each H2A, 2 copies of each H2B, 2 copies of each H3 and 2 copies of each H4. Total 8 histone molecules are present within the nucleosome. H1, you should remember one of the most important thing, H1 protein or the H1 histone protein never present within the nucleosome structure. Normally, H1 proteins lies outside the nucleosome. This black color structure represents the nucleosome. Understand? So here, the H1 proteins lies outside the nucleosome. This is the H1 protein, black color. So this is the H1 protein. H1 proteins lies outside the nucleosome. So what is this? Blue color represents DNA molecule. So the DNA can wrap around the histone proteins and forms the nucleosome. So H1 proteins lies outside the nucleosome. Very, very important bit. So which proteins lies outside the nucleosome, or which protein is absent in the nucleosome, within the nucleosome? H1 protein is absent within the nucleosome, or H1 proteins lies outside the nucleosome. So what are the proteins which are present within the nucleosome? H2A, H2B, H3 and H4. So nucleosome usually consists of 200 base pairs of DNA helix. So this is one nucleosome. So each nucleosome contains how many base pairs? 200 base pairs. Understand? So 200 base pairs are present within the DNA helix. Next. So what is chromatin? Now we are going to discuss about chromatin. So number of nucleosomes can combine together to form chromatin molecule. So what is chromatin? The nucleoprotein fibers present within the nucleus is called chromatin simply. Nucleoprotein fibers present within the nucleus is called chromatin. The chromatin along with nucleosome appears like a beads on a string. Chromatin, it appears like a thin fiber. So the nucleosomes in the chromatin looks like beads on a string. For example, these are the nucleosomes. This is the chromatin. So it is like a beads on a string when viewed under electron microscope. When you see this chromatin along with nucleosome under the electron microscope, how it looks like? Beads on a string. So here the chromosome can be packed. How? First the chromatin can be packed and forms the chromosome. How? The chromatin can be packed. For that they require special type of proteins. They are called non-histone chromosomal proteins. Till now we have discussed how the nucleosome structure is formed. Now chromatin is nothing but it is a thin fiber. It appears like a thin fiber and the chromatin along with nucleosome. So this is the chromatin. These are the nucleosomes. So nucleosomes on the chromatin appears like a beads on a string. Next, the chromatin also coiled and further it condensed and forms the chromosomes that we will discuss now. So chromatin, the nucleoprotein fibers which appears like a thin fiber, okay, is called chromatin. Chromatin can condensed and coiled to form chromosomes. The chromatin can be condensed and coiled to form the chromosomes. For that, they require special type of proteins. How the chromosomes can be formed? Chromosomes can be formed from the chromatin. For that, they require special type of proteins. What are the special type of proteins? Non-histone chromosomal proteins. So, non-histone chromosomal proteins are the special type of proteins which are required for the condensation and coiling of the chromatin to form the chromosomes. Now, chromatin is of mainly two types. So you know that chromosomes are formed from the chromatin. The chromatin itself condensed and coiled to form the chromosomes. For that they require special type of proteins called non-histone chromosomal proteins. Now chromatins are of how many types? 2 types. What are those? Euchromatin and heterochromatin. So what are the 2 types of chromatins? Euchromatin and heterochromatin. So what is the difference between the euchromatin and heterochromatin? There are mainly 3 differences between the euchromatin and heterochromatin. Euchromatin is loosely packed. How they are packed? Loosely. Heterochromatin, tightly packed. Next, lightly stained. Euchromatin is lightly stained. Heterochromatin, darkly stained. Euchromatin is transcriptionally active. Obviously heterochromatin, transcriptionally inactive. So it is loosely packed. Euchromatin, you know that we are discussing about DNA packaging only. Here the euchromatin is loosely packed. Here it is densely packed or tightly packed. It is lightly stained. When you stain with acetocarmine, acetocarmine is one of the chemical which is used to stain the DNA, which may be chromatin or chromosome, whatever it may be. So when you add that chemical acetocarmine, what happens? It is lightly stained. It is heterochromatin will be darkly stained. Next, it is transcriptionally active. Euchromatin is transcriptionally active. Heterochromatin is transcriptionally inactive. What is transcriptionally active? You know that in central dogma of molecular biology we discussed. What is transcription? RNA is synthesized from DNA. It's called transcription. Synthesis of RNA from DNA is called transcription. So here the euchromatin is nothing but DNA. Heterochromatin is also DNA. So here euchromatin only can synthesize the RNA molecule. Heterochromatin cannot synthesize the RNA molecule. So synthesis of RNA from DNA is called transcription. That's why it is transcriptionally active. It is transcriptionally inactive. That is the difference between euchromatin and heterochromatin. With that, we completed DNA package.